Thank you all. We'll be starting in about 12 minutes. So thank you all of you. We are starting in about 9 minutes. Thank you so much.
Hi Prash, can you please unmute yourself?
Yeah, yeah. I'm sorry. It's a forget syndrome, Aparna. I'm sorry. Yeah. So we have about 6 minutes to go. Before we begin, I just have a small announcement that you could probably register for Index Indian Conference on Bioinformatics, which is going to be held between December 4th and 5th. The pre-conference workshops are held between December 1st and 3rd. We have three wonderful workshops. And if you are a life member, all the workshops and the conference is absolutely free. So you're most welcome to attend this particular conference. The conference is available at this index.bioclues.org. Thank you so much, all of you. And I also thank Aparna for a gracious time. We're getting started in about 5 minutes. Thank you so much. So, Aparna, we are starting in about 1 minute, please. Thank you so much for your time. Yeah.
It's okay, Prash. Yes, thank you.
So, welcome all to this Sci'verstion, this edition. This is our 13th Sci'verstion. We are quite honored to have Aparna Banerjee, who is based in Chile in University of Kotola. She has been a women in biology co-chair since last year, along with Sakshi Singh. Aparna is a BSc and MSc graduate from University of Burdwan in West Bengal, and then from Vidyasagar University, and then she went on to do her PhD after getting a junior research fellowship at University of Burdwan. She worked with Rajiv Bandyopadhyay, who is my mentor as well. They have extensively worked on different extremophiles and microbial flora and fauna that have been originating from Antarctica mission from the state and as well as in a Central DST mission project. Aparna is very much interested in the areas of the microbial genomics and how the microbial flora and fauna take up the evolutionary progress and the kind of challenges that she has been instigating ever since. She had a brief a postdoc after she graduated in 2017, and ever since she has been in Chile in working as an assistant professor. So thank you so much, Aparna. Aparna is also quite active in Twitter. She she is absolutely focused on instigating young minds in the microbiology research that she has been doing. She uses a Twitter handle Aparna Microbio, and as well as she uses academic chatter where she has got a lot of friends and colleagues where she takes inspiration from, and also she passes on the kind of motivation and inspiration that she has. Thank you so much, Aparna. Yeah.
Hi everyone, and thank you, Prash, for the nice introduction. It have been like actually it's for me a learning time to be with Bioclues, because I learned many things to how to work in a group, and because this group focuses on mentor-mentee relationship, and that's what I like most, because in my case I realized that how life changes of a researcher when you have a very good mentor who supports you throughout your academic career. So I think I'm very much indebted to Professor Rajiv Bandyopadhyay, because when I look back at me before 2014, I see I was focused, yes, like I was all the time, I wanted to do science, I wanted to do microbiology, but then someone need to take you to a way which way is good for you, and he has been all the time there. I remember that the first cyber session was with Professor P.K. Gupta, and he is such an eminent professor, your mentor, Rajiv sir's mentor, and we are like all having that mentorship with us one after another like this, and I feel so blessed to be in that same lineage. Thank you so much. So Basically, I've done my BSc in biotechnology in 2010, and then I have done my master's in biotechnology in 2012. And then after 2012, for a short time, I did some coaching for the national entrance examinations for PhD, because in India we have to qualify those in order to get fellowship. So after that, I actually gave a lot of interviews. When I remember these days now, I feel like if I'm standing here, I'm really standing here like this. Those times were difficult. These I want to share because many people will be here who are students, and many times they just think maybe that choosing PhD or choosing research is a difficult thing, and what is the way that we should carry forward. So that time I remember I was chosen for interview in 3 IITs. I actually had a small accident at that time also, and if I had like pain all over my body, and then even with that I was going for the interviews in three. Other than that, in some universities, and then I got selected in 3 universities at a time, all are in West Bengal, and I joined one that was also a very good university. So after a year, not even after a year, in 6 months, I started realizing, okay, the PhD is going on, this is fine, but I'm just not liking the work that I am doing. I'm not getting interest in this subject. I was working on plant biotechnology that time. I'm not saying that plant biotechnology is not interesting, but I was somehow feeling like every day I'm doing the same thing. There is nothing, and I feel in research you should have something that you don't know. And I was given with the equations, do this tomorrow and you will get probably these results, and that I was not liking so much. So I just left that PhD in 1 year. And I remember that I didn't have anything at that time. My that qualification national entrance was also coming to the end in 3-4 months that time, and my parents were a little upset about my choice. My father, more specifically, he was thinking that I was in a big university and why I left it. So I again started applying for other position. And that time I remember I got this in Bardhaman University. So Professor Rajiv Bandyopadhyay, he was at that time in BIT Meshta. And I didn't knew that under whom I am starting my PhD, because it was an interview with the previously recruited professor of Bardhaman University. And then the result came out, so I am allotted with Professor Rajiv Bandyopadhyay. And I remember like how a mentor should be. I joined at 9th December 2014, and he was there. He just joined at Bardhaman University that time. Why I shared this? Because I have seen a lot of mentors in my life, a lot of professors, but there's a very few who really think of the student. And he probably came from BIT Meshta, and he had some work to return to Ranchi at the same time, and we needed the application letter in 3 copies, and there was some problem of the Xerox machine, and he told, Aparna, you just write 3 copies. I'm first signing it to you so that you can join by today, because we have 3 days, but I don't want you to join more late. You can join today only if you can finish writing the 3 letters. And that's so much helpful. And I also remember, because we didn't knew each other both, he told me, let me give my introduction to you. And I heard his stories. That was like the first key point in my life. Well, one professor is giving his introduction to me, what he has done, and I was feeling motivated. The inside of me was like, well, so I can go to Antarctic expedition. Well, so I can go to South Korea. I can also go to US one day. And that's the way he was like weaving the dreams in a student's mind. And the rest was all very good experience after that. I almost didn't have any trouble after that. I have shared here some of the photos. One was in 2016 probably, that I was selected in Young Researcher Forum in Thailand. So that was a very changing moment in my life, because I was going to go alone to Thailand. And in a middle class Bengali family, it was like, you will go alone, how you can go? But then I managed, and my parents are very supportive in a way for me all my life. I have put my parents' picture also, because whatever I am here today is because of them. So they should be here and people should see them. And in Thailand, I actually for the first time realized how the international researchers are, and how they with what confidence they talk actually. So from there in 2017, 2016, this was in 17, I got the best oral presentation in West Bengal State Science Congress. In West Bengal State Science Congress, they have some regional groups. You get selected from there, then you go to the state, and then Sir Rajiv sir all the time used to boost us to apply for the Young Scientist Award of National Academy of Sciences India or Indian Science Congress. So in 17, I applied for National Academy of Science India. I got there the Swarna Jayanti Puruskar for best presentation. And then in 18, I got Indian Science Congress Association Young Scientist Award. That was on new biology section. And then there is a photo that I am taking the award from Najma Heptullah madam, Dr. Najma Heptullah. And then in 2018 June, I was awarded with my PhD in 29 June. And in 18, I was at the same time I was applying for the postdocs, and in Chile I got the first postdoc position with Professor Sara Cuadros. And the way I got the postdoc, that's also very interesting for me, because normally we go for the traditional way, we apply over the mail, and then many of the times we don't get their response. So I wrote to her over Twitter, and she was very gracious to write to me that, look, there is a position coming in 3-4 more months. If you are interested, I can share with you the position. And that's the way I came to Chile after getting selected to that position. So in August 2018, I came to Chile for the postdoc, and there I was in the postdoc for 1 year, and then I win one funding in Chile. It's Fondesit initiation, and with that funding, I was promoted to assistant professorship. So from that onwards, now I'm working on that project only. Basically, I'm a microbiologist, because if you can see, my bachelor's project was on, though these are very little experiences in life, but that likes make the path of a student. I realized that I really like microbiology. When I look back, my bachelor's degree project was in a biotech company, and it was on erythromycin resistant gram-negative bacteria. So from there, I did my master's project in drug-resistant bacteria for urinary tract infection, and specifically some Fimbri and Pili-related genes. Then my PhD was in extreme environment, microbiology of hot spring, and exopolysaccharide production. And I just so much like that work. I really like biofilms, extreme environment, microbiology. I realized that this is the part that I like. So I was focused that in my postdoc, I want to do the same kind of research. Maybe the environment can change, but I realized that there is so much to do for the environment that are unexplored, where there have been no work earlier. The unexplored environment can be many things, not only hot spring. For example, Arctic Ocean, Antarctic regions, Soda Lake, like Lonar in Maharashtra, then the high altitude lakes in Himalaya, or the geysers, or many nuclear contaminated site, acid mine regions, many. And most of the times, these are all the environments where the life does not survive in a way that we think, like the animals or the plants. They don't survive in that kind of region all the time, but microbes do survive. And I realized that in the traditional conservation that we talk about is so much focused on plant and animal conservation. The least we think about is microbial conservation. I will talk more about that later, but I really started liking that work. And in Chile also, I am working on actually Chile have all over, it's covered with Andes mountain, and we are working in this project. We are focused on the microbiology and how they survive polysaccharide production biofilm of central Andean mountain region. So I feel that if science is your passion, there's nothing in this world that can hold you back. I will share one of the story. I started working during my PhD with 6 isolates, I remember. And out of that six, one was very interesting. It was producing very good amount of polysaccharide. So after the sequencing, we came to know that this is a Bacillus anthracis. And then what came to our mind is Bacillus anthracis is a pathogenic bacteria, because in Prescott you all the time see like Bacillus anthracis is an obligatory pathogen. So at that point, I remember that Rajiv sir told that we thought that we have to stop the war. But he asked me, Aparna, do you have the DNA with you? And I told yes. So at least we can do the whole genome sequencing. And let's see why, if it's pathogenic, why nothing happened. And I was also thinking, if it's really that pathogenic, why nothing had happened to me? Because I never thought that in extreme environment there can be pathogenic bacteria also. There are reports, a very few reports, when it is contaminated with like human faces or this kind of things. But where I was taking the sample, Panifala hot spring near my city, this was a barely human oriented region, and it was in one coal mine region, very isolated from the locality. So it has less chances to be pathogenic. So when the genome result came, we saw all the features or footprints that this bacteria can be a non-pathogenic bacteria. So in that time when I saw this is a non-pathogenic bacteria, so we worked more on, completed the work on polysaccharide, and this has been the first report of exopolysaccharide characterization from Bacillus anthracis. There have been reports that Bacillus anthracis produce EPS, but this was the first report of exopolysaccharide from Bacillus anthracis, and it was just showing the same type of polysaccharide that the Bacillus group produced, dextran type of polysaccharide. And dextran is highly useful in pharmaceutical industry, in food industry these days. But then there was again that factor that it is pathogenic. If it is pathogenic, then how we can like, there is a question of the pathogenicity. So at that time, we got in touch with Professor Rakesh Bhatnagar from JNU, and he has been, he is one of the world leading authority in anthrax research. And I remember that he was also excited about this, because in his research life, he has never seen something like this, that one Bacillus anthracis that is non-pathogenic. And so I went to JNU for a brief time, for 1 month initially, and then another 1 month, and we did some genomic study more in depth. That was the time like I moved in bioinformatics. I was not a I did biotechnology, I knew bioinformatics, but I never utilized that in my research. But when this was the time that I shifted to microbial genomics, and then comparative genomic study, pangenomics, that time I started getting interest on that, and I started learning by myself. And so we have some very good couple of results on pathogenicity related study with the traditional anthrax causing Bacillus anthracis, by MTT assay, by in animal cell line, in mice model, and in many others. So what we get from these, that this Bacillus anthracis is a really a virulent variant of Bacillus anthracis that is reported from hot spring. And there was earlier only one report of a Bacillus anthracis from one Algerian salt lake, soda lake. So there was no detailed report of this bacteria though. So this was in this research for me was unique in many ways, because I still remember that in Prescott it was written Bacillus anthracis is an obligatory pathogen. So in science there is like nothing you can say this is this. This is final. There is everything that we can get from there. So there are more works on polysaccharide you can find on ResearchGate. But I will go on what I feel personally about microbiology and about microbial conservation. So when Prash, can we go to the next slide, please?
Yeah, please. Yeah.
Yes. So there is just a little mistake. Microbes too, TOO. Microbes too need conservation. I so much believe that our planet belongs to all, and microbes too need conservation, like the plants and animals. And we need to talk often about that. Microbial conservation do matters. So there was a time I was thinking that why in the conservation there have been no report of microbial conservation. All the time that we talk is about the endophytes or it's about the symbiotic bacteria. What about the others? The others are also have a big ecological role they are playing in biogeochemical cycle and in their own ecosystem. If we hamper that ecosystem, if we just go and take bath in a hot spring, we are just destroying the million years of footprints of bacteria that was surviving over there. Because if we think like the earth 3 million years ago, the earth was bubbling hot, and what was surviving over there other than bacteria or archaea? Nothing. So the hot spring, they are basically taking the same footprint over the years. Not only about hot spring, any extreme environment where there is very less human exposure, and they started we started invading their own ecosystem. What we will lose is their footprint, and maybe we will never know what those bacteria was helping in that particular ecosystem. So I was just checking at some point of time what had been the publication in microbial conservation. There was the first publication in 1964, and that was actually the first publication from where the culture collection idea started. This was one very interesting publication for me, because though not all the microorganism are cultivable microorganism, and actually 1% microbiome are cultivable, but even the thought of microbial conservation started. Then in 2019, this paper for me was very interesting, because there they were talking really about the necessity of microbial conservation, but this was more focused on the host associated microbiota that I was again talking, that we all the time think that microorganism, if they are helping in some way to the human, or microorganism if they are helping in some way to the plants, like all the time the host associated microbiota. What about the others? So that time this idea came of, and we wrote this letter about the emergent climate change impact throughout the world, and how there is a possibility that we lose the microbial footprint, and why it's needed to talk about microbial conservation at this point of time. So here is some picture of the extreme environment that I have worked. The first one is Panifala hot spring, and that I have done during my PhD. I remember Rajiv sir told me that, Aparna, you can do something from your city. You can do something for your locality. And at that point of time, I thought I was basically working on Southern Ocean microorganism before that in my PhD for like 4 to 6 months. And then I thought I was checking what is so unique near my locality. And then I suddenly see, there is a hot spring that is not in my city, but actually far from my city, like 1.5 hours. But I can really go and see how it is, because my idea at that point of time as a student was like, hot spring is like Bakreshwar or Monikaran, people take bath there. This was my only idea about hot spring. So I go there, and it was very interesting. The water was like 66 degrees centigrade, and of course then later on we went in various season, and the temperature changes according to the season. But it was very interesting and very isolated for me, that place. And Professor Rajiv Bandyopadhyay also went to Southern Ocean expedition in 2011. So I also worked on some of the Southern Ocean isolates and their exopolysaccharide production. And then now I am continuing in the hot spring of Andean mountains in Chile that are unexplored. And the two kids, the photos of they are working with me for their thesis of biotechnology. They are my students, and they are working in this Fondesit initiation project, and they are very interested. They most of the time tell, because now of this, for this pandemic, we the students are really not allowed at this time to go to the laboratory, but they all the time write to me when they can again start working in the laboratory. And I feel that's so good when I see this in a student, that they really want to do something. They don't know maybe what to do, but when I see that they really want to do something, I feel like myself 10 years back, that I badly wanted to do PhD. I didn't know what is the way, but I was just going because I really needed that doctorate in front of my name. I remember the reason was, there is Mrs. or Mr. The woman is always someone's Mrs., but doctor is a suffix, a prefix over our name. that is not gender biased, and that is what we need in the world, that men and women are really counted as equal, because both the gender contribute to the society. And so can I go to the next slide?
Yeah, yeah.
And I will just end up with two of very close thing to my heart. This is in Bardwan University, the first picture of Jagadish Chandra Bose. And I remember one incident from here. We used to work in the third floor. Our laboratory was in the third floor, and one middle-aged man every day evening used to have evening walk in front of our department, and in front of Jagadish Chandra Bose statue, he used to open his shoe and used to make like this, pranam. And that's for me, and my Rajiv sir also have seen that actually. We all the labmate used to discuss this, that one common person, maybe who just loves science, for him Jagadish Chandra Bose is near to God, because he discovered something that changed the world's idea about life, that plant to communicate, plant to have life. And this is actually written in the back of Jagadish Chandra Bose statue, and it's a famous quote of him. And I so much believe this: the true laboratory is the mind, where behind illusions we uncover the laws of truth. So this is the temple, and we need to pray this, and we need to uncover the truth more by thinking process. There is so much things that is hidden. And then this is a poetry by Robert Frost. I remember I was in class 11, and this is a poetry that made a big impact on me. These two are the quotes that I all the time remember in my life when I feel something is not going okay, or something I can change in my life, or like how to get motivated. This is so good. The woods are lovely, dark and deep, but I have promises to keep, and miles to go before I sleep, and miles to go before I sleep. So there is no end of path. We have to continue walking no matter what difficulty comes. Every day there comes some difficulty. When I have been in Chile, the initial time I came, I had the idea, well, I'm going to South America and maybe people talk English. I came and I realized no one talks English. They speak Spanish. And I minimum take 1.5 years without even understanding what they are saying. Or I used to communicate, but I felt I get a lot of good help and wishes from the Chileans, from the Latin Americans. And today I speak, it's like a medium level speaking. I think I'm still speaking like 30% Spanish, but I speak. I am learning at this age one new language, and I like this. I like listening to Spanish music, because you cannot survive in another country without learning their emotion, without learning that language. So that's I'm learning for myself. That's I'm learning so that I can live in this country. And I felt all the time good vibes here. People are very kind, considerate, and the university, I feel like a family. All the time that I felt some problem, I went to the higher authority. They have tried to solve my problems. Problems do come, and at some point of time, maybe I thought, well, I will go back to India no matter what. I cannot survive more. No, no, no. But then there have been something good. I remember one quote of one of my teacher of English when I was in 8th standard. I was very depressed for something, very little thing maybe. I remember I 2 times fall from my bicycle, and I was saying that I don't like to come. I have so much pain, and these I was complaining about life when I was just in my 8th standard. And he told me one thing: Aparna, tell me how many times you get benefited from someone and how many times you got betrayed. And I was counting how many times I got betrayed, how many times I got benefited, and I was really not able to count how many times I got betrayed. Maybe I told, maybe 2 times. And how many times you got benefited? I can't remember, sir. And he told, look, you need to believe on people, you need to believe on life, because life will go on, and there are still good people, there are still good things in the earth that you should get excited for. So there have been so many mentors in life that changes my life. I remember one of my professor of biology. She was a nurse, and she was living near my house, just one house after me. And I used to go to her just to learn biology, like, look, sister, I don't understand this part, can you, it was very like very homely kind of thing. And she has a big impact on me for loving biology. My mother have a big impact on me. She is not like she's from a village of West Bengal, and she don't have education like all of us, masters and PhD, but she has been with me during all my journey. Being a single woman coming from India to Chile, she was the one who stood like pillar by me. There has been so many times when I thought in department there has been some problem or something. My PhD supervisor, he has stood with all of us like pillars, and whatever we all are doing is because of him. And then when I remember that, how all the time my father told me, Baba, I don't like this, Baba, this person is bad, Baba, this is like this, Baba, don't do so much complain about life, you do your work and life will get back to you in a good way. So this is the way I learn from people. I actually learning a lot from Prash now, how to manage a lot of works with so much passion and calmness. So there's not a single point when we should stop learning. And science is all about these questions and the passion for learning. So I will like stop here and I will wait for Prash if we can discuss on something else.
Uh, this was an excellent talk, Aparna. Thank you so much, for the wonderful inspiration and motivation. It's been absolutely amazing. Friends, one thing I really wanted to tell about Aparna. She's so nice that she was nominated for Bioclues Innovation Research Development Award last year by Rajiv sir, and she was not through. In fact, we didn't chose anyone, but she was so nice. I mean, believe me, that after the interview ended and after we announced that we're not going to give away awards to anyone, she sent us a message that I didn't apply for getting the award, or neither do I expect any awards nor promotions. But I'm here, for serving the young brethren, and whatever I've really learned, through my mentors, and that was none other than Rajiv sir. I mean, you see, you find, very few people like Aparna who are absolutely, always smiling, always, down to earth, always, helping, always mesmerizing, always, replying, dot on time, doing the tasks and all those kind of things. And if you really see her wonderful quotes, miles to go before I sleep from Robert Frost, that speaks, volumes about her, wonderful passion, that she has instigated. Thank you so much, Aparna, for your wonderful time. Yeah.
Thank you, Prash.
I have several questions, Aparna. To start with, I would probably ask you first. I would say gracious. Thank you so much. And the second most thing, Aparna, you have extensively worked on polysaccharides, and absolutely, polysaccharides are easily degradable. They make sense, for the wonderful evolutionary entity towards nanoparticles, and they could be, wonderfully poised, towards, amenable drug targets, and probably towards, theranostic, drug targets. So, post-COVID times, how do you see the production of these polysaccharides, coming from these bacteria?
There is actually, Prash, all the bacteria, they mostly produce different kind of polysaccharides, and it's very difficult to characterize them from a biochemist point of view, because most of the times they have some substituted sulfate groups or some uronic acids or some rare sugars. So there are some polysaccharides that we know and that are patented, like dextran, xanthan, levan, gelan, cardlan, succinoglycan, a lot. So they are like get regularly used. But for example, marine polysaccharides are normally, the marine polysaccharides are the one that are therapeutically used in pharmaceutical industry as drug carriers. because they are sulfated polysaccharides, and sulfated polysaccharides are have been there have been a lot of research that they have good therapeutic properties. So marine polysaccharides are the polysaccharides that have this kind of property. But the thing is, what I have seen during when I was working with the exopolysaccharides of the Southern Ocean sample, what happens is like over 6 months their production changes, their characteristic changes, and the isolate that looks 6 months back after 6 months doesn't look the same. So I talked with two person. One was Dr. Pradipto sir from Bardwan University. He's a great microbiologist. He told this, that Aparna, the characteristic of the bacteria do change. They accumulate mutations, and you have to think that the temperature or the pH or the things that you are making for the cultivation of the polysaccharide is not the same that they were in that particular environment. You cannot mimic it completely. And another time I had the same like opinion from another great scientist in Chile, Dr. Jenny Blemmy. She has a lot of work on one hot spring of Antarctica called Deception Island, and she told the same, that she has faced the same experience. So this is very important, that for some extreme environment, mostly the bacterial polysaccharide that are till now patented and are getting used regularly, I have seen that not like completely, but mostly they are lactic acid bacteria and soil inhabiting bacteria. Very less are actually psychrophiles or thermophiles. The reason is mimicking the environment that they were living. But we need to actually work more on that, and I really don't know some good answer to this, like if extreme polysaccharide from extremophilic bacteria like can be readily used for a long time. There are reports, but fewer. But definitely polysaccharide produced by the soil inhabiting bacteria or the lactic acid bacteria, they are regularly getting used on that. One of the very good example is dextran and xanthan. Both dextran is a neutral pH polysaccharide. Then that is the reason normally the polysaccharide are acidophilic and they are acidic in pH. But dextran produced by the lactic acid bacteria, and mostly bacillus group, not only lactic acid bacteria, sometimes geobacillus also. So they are the one that is mostly used as blood substitute, as drug carrier. And we have also worked on dextran type of polysaccharide from Bacillus anthracis. So there's a lot of possibility that they can be used as drug carrier. And so this is what I think, that there is a big possibility, but well, a lot of research is needed on that as polysaccharide as commercial drug carrier. But they are regularly getting used. Like for example, succinoglycan is a polysaccharide produced by the soil inhabiting bacteria. If we see the cosmetic creams, sometimes in the back, if we see their composition, succinoglycan is there. If we see some cookies, some good cookies or some jelly kind of thing, we can see xanthan as one of the compound that is produced by Xanthomonas campestris group of bacteria. So we don't know, but actually everyday life we are using bacterial polysaccharide. So when I say people, bacterial carbohydrate, really? And I say, we use it in everyday life. Cream and cookies that we take every day. And succinoglycan and xanthan are very two common polysaccharides that we eat every day in our regular life, maybe. Prash, it is muted.
Yes, yes. Can you hear me now? Yeah.
Yes. No.
Yeah, I'm sorry, I changed, the background. I was just trying to make an experiment, and somehow, this popped up. This is very interesting, Aparna. I know, when you landed in Chile, you might have, had, some initial acclimatization problems. So how did you overcome these kind of things? I mean, I know I could also see from your tweets that you send tweets in Spanish as well, in Espanol as well. So how did this come up? Yeah.
That come up suddenly probably. I was like all the time with communities of the university. My initial day 6 months, I was very vulnerable that time. If I look back myself at that time, that is so much not Aparna Banerjee. I am a very positive person, happy person, and thinks about, well, I can do anything, like I can go at any part of the world. But that initial 1 year was like the most difficult days of my life. I used to feel depressed many times. I will not call the word depression because that's a very big word, but I used to feel sad. Like all the time, I used to complain about my life. And if I have some complaint, I call Rajiv sir, I call my parents. And one day, sir told me, Aparna, coming back is easy, but try to see how long you can survive. That is important. So there was one time we was organizing one seminar in last December 2019 in Chile. We got a fund of that with my ex postdoctoral supervisor, and we invited Rajiv sir over there. And I was talking very little Spanish, and what he told me was, "Ah, Aparna, you started speaking Spanish. How he all the time boost up the students like that, even if they do some very little things, and we get motivated." So I was that time I thought that, look, if I have to survive over here, I have to speak this language. If I My only knowledge about Spanish was gracias, hola, buenos dias, and despacito. This music is popular in India also. But then the life changing moment was when I got this funding. That's almost 79 lakh in Indian money. And the funding was big. I was dreaming big. There was a lot of things to explore that was new, that was unexplored earlier. So I really was motivated that time to do this. That this country has believed on me, has given me a project, has honored what I have believed, has honored my idea, most importantly. So I have to do something for this country, have to do something for science, because Chile for me is like one of the model environment to study extremophiles. If you see the north, it's Atacama Desert. If you see the south, it's Antarctic environment. Atacama Desert is also the driest point of Earth. The entire Chile have high UV exposure. There are a lot of hot spring, geysers, soda lakes, salars. So I was really excited at that point of time, and that was the time maybe I don't remember what was the module for me, but I started working on myself at one point. I remember at the night I used to write what are the three best thing happened with me at the day. I was alone. I was not in an Indian community for the first 1 year. Then slowly more Indians started coming. Now I have a lot of friends also, but that point was not there. Then I increased that. Five best thing happened with me in the day. 10 best thing happened with me in the day. And when before sleeping you think of the positive thing that happened to you, you really feel, okay, life is good, I can survive. So that was the change of time. And then I started getting more Chilean friends, and they started speaking with me in English, helping me. A lot of Chileans helped me, actually. I cannot count even them. There are so many. So that's how the changes started coming. They started forwarding me, listen to this music, maybe this will fit your choice. So that's the way I started liking Spanish. So when you like a music, you try to feel what is the meaning of that, and I started seeing that. And I also get very good warm welcome from the old people of Chile, like, you are from India, so far, India is such a good country. And let me tell you a very interesting information. I think there was a serial called Saraswati Chandra in India. So that serial in Chile, in Chilean national channel, they translate it in Spanish and they see it. And everyone knows Saras and Kumud. And that's the way they portray Indian culture. And they are so welcoming and positive about India, though it's like 17,593 kilometers from my originating city, Asansol, to Talca. It's a big distance, but I feel good nowadays. Not so difficult.
Amazing, Aparna. I mean, well said. Yeah. So, Aparna, Rajiv Bandyopadhyay, sir, is absolutely a phenomenal person. Is absolutely enterprising. He was also my mentor. He's also my mentor. In fact, we cherish wonderful relationships with him. When I was in P.K. Gupta's lab for a very short stint, his helping was absolutely immense. I mean, if I really see the commonalities between Rajiv sir as a mentor, and you yourself as a wonderful, young scientist today. There is one thing that is common between you two. You always keep smiling. You always think positive. You always are helping. How big was your or how big was the kind of influence on him? You know, what is the kind of inspiration that you have really taken? Or learning lessons from him.
I feel emotional talking on this. And I remember one thing he told me the day I was leaving India and I was coming to Chile. Aparna, you are very emotional and keep that in control. I remember in my pre-PhD, my cat is coming. One moment.
Oh yeah, very well. Oh, what's the name of her? Yeah,
Her name is Lichi. Lichu.
Ichu. Oh, wonderful. Oh, we love pets. We didn't know that, Aparna. Good.
And
Yeah.
she bomb up all the time when I'm in presentation. She likes computer. So I in
Oh,
my
wonderful.
PhD presentation, I put like I cried at the last because I felt so hard that I'm leaving this laboratory. I have to start my new life now. I have to be alone. Like I don't have a investigator. Don't have a supervisor over my head. And obviously I started feeling that time. Sir also told that all the time you will not have supervisor. Now you have to start thinking of your own. But he has been a mentor. This is a difference between a PhD supervisor and a mentor. When I look at some point of time, some of my friends who has done their PhD and they speak negative about their supervisor, I never actually cannot imagine that how amazing Rajiv sir had been. One example I told that there was another example when I was coming to Chile, and he has been like trying his best to help me in a way. I was going to lab less even after when I was like applying for the visa and everything. He was telling me how the way I should survive there. What are the things that I should keep in even my luggage? Like he was sharing his experience in United States, and then he came to Chile for last year, and then even after coming here, he cooked one day khichuri for us, for my friends and me. So this is a person Rajiv sir is. I cannot explain in words how he is. And he's so all the time in replying. If you just mail him once, he will definitely reply you. This is the thing that I learned from him. Like you have to think your students or your colleagues and family. Here is another lab member from my lab, actually, Srobona. She has been also absolutely amazing, very positive person, and just like taking the legacy of Rajiv sir. This is actually in common in our lab. I think most of the person there, we just don't say that sir as sir. This word sir has been such a big impact on me. And when I actually met Professor Bharsne from PK Gupta's lab, PK Gupta sir himself, you, a lot of people other than you, because I also worked at some point of time with Rajiv Bharsne sir. So I know how these people are. They all have one common characteristics. They are extremely helpful and they think about the students first and then about themselves. And they keep, like, one very interesting thing of Rajiv sir, keep writing, practice. That all the time used to say. Gupta sir also used to say, practice writing, be on science, come to the lab daily. So this was a very important thing for Rajiv sir. Come lab early in the morning. There was time 9 to 9. Have been in the lab for 12 hours. Saturday also, then Saturday in the evening, I used to take a train, come to the home, and then Monday again used to go back. But I used to love laboratory. That have been the time that if someone tell me, God give me one chance, what is the thing you want to back in life? I want to be back in my PhD. That was the most amazing time. And this is a thing, another thing, in Young Scientist Award, for example, we know in ISCA Young Scientist Award, we need to apply as only the author. There is no supervisor with you. So I was during my PhD at that time, and he was the person who motivated me to apply for this award alone, not as poster presentation. Indian Science Congress award, Young Scientist Award, and I was one of the youngest people over there, because I think the highest age is 32, and when I got, I was 27. So I was during my PhD, I was not doctorate, and I win that award. And I really never hoped I will do this, but I anyhow managed it, and all the credit goes to him. Whatever I'm here today is because of my parents and Rajiv sir.
Wonderful, Aparna. I mean, that shows, the wonderful inspiration that you have taken from Rajiv Bandyopadhyay sir and your parents. I know it means a lot, for you. You have got, wonderful, upbringing, through your parents, and of course, more towards the wonderful mentorship over a decade from Rajiv. But one thing, Aparna, there's also a kind of a strong what you call, motive force, within you as well. A strong, willingness, to do something, which is always,
Yes.
appreciated.
I saw Prash that I saw many PhD students who think of starting PhD, and that at some point of time they just lose the interest. They don't feel to write paper anymore. They are complaining, we will only make publication at 5 impact factor. No need to write small papers. I just feel, what is the run of this impact factor? Who decide impact factor? In mathematics, even getting one impact factor is difficult. In extremophiles, 2.5 is the highest, that is impact factor of the journal itself, extremophiles. So, and people just lose interest. I still feel myself when I have students, I try to behave with them in their age, and I feel motivated all the time seeing the students. So this is important, like having interest on whatever you do, science or whatever. You look at yourself
Wonderful.
all the time, Prash. You are like, if I write to Prash, I will get a reply in 10 minutes, I know. And that's how amazing, Prash, you are. And you are helping the young people. That's most important.
Thank you. Thank you for your kind words, Aparna. I mean, I know, coming back to your the words, on impact factor. I know, these metrics, doesn't really make sense. What kind of, essence, how good, the science is really, moving forward, with the impact that you make matters a lot. So moving forward, Aparna, I mean that anthrax story is absolutely inspiring. I think you might have really felt like that you won a Nobel Prize. Looking into something called a non-pathogenic one, that from a Bacillus anthracis species is something, something very mesmerizing. So if you really look back, Aparna, do you see? I mean, can you visualize? Maybe just because you have got this isolate, from a hot spring, that from a non-pathogenic source. Do you see that probably the hot springs, the kind of the microbial flora that everyone takes into account? Maybe every people across different hot springs across the world, they probably bathe. They get into the water. Do you see that there's something the human microbiota also involved? Maybe thus making these pathogenic convert themselves to non-pathogenic ones. Do you sense something that such things might really happen? Because it's other way around. We might have known many non-pathogenic bacteria turning themselves towards pathogenic bacteria. That is well known. But the other way around, this is something different. Maybe who knows? If you really look into the evolutionary cascades of the non-pathogenic isolate which you have taken from the hot bath or the hot springs. So, how do you visualize as a microbiologist on this particular episode, Aparna? What are your insights?
These actually, these that you told, me and Rajiv sir also asked me the same. What do you think? What is the way? The top approach or the bottom approach? I don't know. The word that I all the time felt it, my intuitions are they have been non-pathogenic all the time, because without the human intervention, without the introduction of antibiotic, for me, there is no reason that antibiotic resistance can occur, or a pathogenic bacteria or pathogenicity island can occur. For me, they have been non-pathogenic throughout the way, and with the time, in order to survive against the environment, they had the pathogenicity incorporated within them. This is what I feel. But obviously, the opposite approach can also happen. I remember I applied in Newton Bhabha Fellowship that I was not selected on, with the first person who worked on Bacillus anthracis genome from Cardiff University, and he was really excited to work on this part, but anyhow I was not selected on that. So with the prophage analysis we can really get on the, I have done prophage analysis but through bioinformatics approach, but if we could have really worked on bacilliphage and can see how the changes are going on in the cell after introducing the phage in the bacteria. So that was like a clue where we can get this idea. But I'm really open to doing this research back. Even we can do something with bioinformatics. The genome is available, the data are available, and if in some way we can see this. I really don't know, Prash, if through bioinformatics we can do this, but I really want to do. The strain is also deposited in the culture collection. So at later some point of time I really want to resume this and get an answer to this work is something that is like the best thing I have done in my life. So I really want to do that in some point of time in my life.
Yeah, wonderful, Aparna. I mean, if you ask me, I've extensively worked on these particular domains. I mean, we have identified, certain domains, that are absolutely pathogenic, and many of
I'm going
these.
to write to you after this now.
Yeah, no, for sure. In fact, I need to tell you one thing which you probably don't know. When I started my probably PhD in 2004, or when I was, trying to make an attempt to get into my PhD in 2003. Uh, I discussed a lot of things with Rajiv sir as well. That's because a lot of signatures do exist for pathogenic bacteria. So if you really see, if you really take the pathogenic bacteria and non-pathogenic bacteria, there are a lot of common domains and motifs. There are some signature sequences that are absolutely considered all through the evolution, and eventually owing to the changes that might really happen through the evolution, we might even find a lot of non-pathogenic bacteria, turning themselves towards pathogenic bacteria. And that is where we have done a lot of studies. We even published a couple of papers, also the one with Rajiv sir as well, way back in 2005. But my interest was more towards human genomics, and that was how I moved towards that. Although I did my BSc in microbiology, I steadfastly put up my interest in human genomics. So moving further up and now. I mean, getting something like 80 lakhs grant is not a kind of an easy task. That too at the helm of your early career stage. So how good was your feelings? How difficult was you to get this particular grant? And I know there's a lot of innovation when we really talk about you know your grant. So what are your feelings? And can you please tell or enlighten the young audience here how to get a kind of a winning grant?
I For me the winning grant is like your hypothesis need to be strong. When I got the result, I really didn't knew I will get it, but I wrote, I help writing funds when I was in my PhD. All the PhD students do that actually, helping writing to their professors. But I was not like really knew how to do it for myself. The idea was there that I know that this region have a lot of unexplored hot springs and I want to see the polysaccharides and some novel polysaccharides that can be applied in food industry as the food additives like dextran or xanthan. I wanted to search for some novel polysaccharides because there is a big gap. If you see the patent analysis, the top countries are Russia, China, United States, Canada, some very little India, and part of Australia. Actually mostly dominated by China, Russia, and United States. So I was at that point of time when I looked at the hypothesis. My hypothesis was strong. I really knew what I want to get. I wanted to keep my objective small. I really don't wanted to do promised a lot of things. My objective was very fine. I want to see first isolating the bacteria, getting the polysaccharides, and then trying to find out their properties as food additives. So these were my finest point of objectives. And the objectives, this is a common objective that people find, that people like frame in their way. I think this was a easy way to get the fund. And the another thing was, in my materials and methods, I was very clear what I am going to do. Many of the times what we feel is that we can do that, but we lack the material methods how to do that. Because I already had the idea of EPS, it was really easy for me to frame the material methods. all the work that I have done, all the supporting papers, how the way to do it, and I explained it all in a small paragraphs, and like support it with the publications that I want to do, something that I want to do, for example, new. And then making the maps of the study site is also very important for me. Whatever I could not do it at that time, but what I did, I put the latitude and longitude of the position and the site, so I make a table. And it's not like, many times I have seen people writing fund, we will collect sample from there. Now the reviewer don't have so much time to go and write all the details there. No, make a table and tabulate it. This you are going to choose. These are the interesting character of them. I feel this was interesting. And then when I got the reviewer's response, there was like, in Chile there is a point of English because it's a Spanish speaking country, so that was a positive point for me because my English was comparatively fine. And the second thing I remembered, the second reviewer told to me that she should get a fund because she is a young researcher. And that's a very good point here, that here the young researchers are really appreciated for their idea. So I really didn't had a hope. I applied at that time, but I thought if I get the like the comments back, I will make it better, and next year again I am going to apply. But I got it that year only, and now the second year already started, and we have got the fund of second year, and now we are trying to buy equipment with fluorescence, luminescence, and making the absorbance of the equipment. So we are like, that's the way I get it. And these are some of the points that I feel that it should be clear, the objectives, hypothesis, and the materials and methods point by point. So you should be very clear what project you are writing, not something like it's flowing in the wind, not something like that. You should have a base on that.
No, wonderful. Well said, Aparna. Moving forward, Aparna, if you really look into the microbiology research, I mean, seldom we find people taking up microbiology. So what is the best advice especially to the women researchers who would really take up microbiology? Because I don't see the general microbiology really considered as a kind of a good household subject or a commercial subject these days. People are swiftly moving towards medical microbiology and all those kind of so-called catchy phrases, catchy terms. So what is the best advice that you would probably give it to the young audience?
I think the best advice that I can give is take any book of microbiology, any good book, can be Prescott, can be Black, can be Tortora. Just read it from the first to the end. If I remember my bachelor's degree in biotechnology, there are many books that I don't only go from the back and the syllabus. I completed Lenin's, I completed Karp, I completed Prescott, like storybooks, first to the end. And if you go to these books, what we don't study is that much extremophiles. What we don't study that much are the endophytes. But look at the research. People are, when we leave the subject and we come to research, so many research are going on that. So in order to get really interest in the subject, take any book that you like, any microbiology book. And just, it's better to have something like printed, colorful, because I'm not so like tech savvy person who like to read books in mobile. No, I really prefer one physical book. So I really got interested on that. And the second thing is get some notification from Science, from Nature Microbiology, and get their updated papers and read that. For example, very few people maybe know that there have been 1 month back, there was a publication in Science about a microbial hotspot in Mexican lake. And this is something a path-breaking research, because there have been earlier one report on this kind of report on Yellowstone National Park. There have been one report of Atacama Desert. So this has been one such kind of paper, Mexican hot spring, Mexican lake as a microbial hotspot. But very people actually know about that. We know so much about the Atacama Desert, the Yellowstone National Park. The paper came in Science, but very few people know it. And I don't saw even in Twitter, people are sharing so much on that. I really don't know why. But this is the way we can keep ourselves updated. And the best way is to be active on social network, not just to chat, but to get the updated news, because all National Geographic, Discovery, or these big publication house, they are all there in Facebook, Twitter, Instagram, and they are so active. They post it every time. So I like to get myself updated over this. That's one of the reason I do social networking side. And that's the way I think people can get interest on microbiology or even in bioinformatics. I think women in bioinformatics, Prash, is very less in India. Bioinformatics is a field that is mostly dominated by male, and a lot of women is needed there to code it, to do data science. So I feel that this is the way we should keep ourselves updated.
Wonderful. Always positive and inspiring, Aparna. Thank you so much. The last few questions I have selected, one question, from the audience. Anil, who is also a professor at Vignan University, he asks how to decrease the solubility of carbohydrates without adding carriers.
Ah, I don't think it's decrease the solubility, but the problem in generally in polysaccharide is the solubility. So one point can be is the first thing is obviously we just cannot make the production without purification. So we have to go for trichloroacetic acid TCA precipitation or the other acid precipitation. The second thing is doing the lyophilization and then getting the powder. Even after that, many times I was seeing, because I was working on a polysaccharide that was also contaminated with pigments. So the problem was how to isolate the pigment and polysaccharide apart. So that time I used some organic solvent to separate it. But another point, even after that also many times they are not so much soluble. So the one way can be heating them, slowly heating them, these increase their solubility. So this is a suggestion that can be done. I personally do that when I need to get the solubility, heating them slowly.
Yeah, wonderful, Aparna. Thank you so much, Aparna, for your wonderful time. Before we say goodbye and I give you one final vote of thanks, do you have any comments, Aparna?
No,
Yeah,
I
yeah.
feel so good. Actually, there have been so many questions here, and I felt so good. Shoma, I think I was talking with Shoma a few days back. She's from Kolkata, and she was really interested to join Bioclues because she felt the same, that bioinformatics in India there have been not a lot of women, and I really, this is the way we help. We get help when we talk to people and we share our experience, and we get more people joined in the group. And I feel so great on that women are taking more interest and privileges to coming forward, and I feel good on that the society is changing, that the world is changing.
Wonderful, Aparna. I think
I think,
Jeraman sir has got to say. Yeah, please, sir.
hello,
Yeah, yes, sir, please.
hello, yeah, hi, yeah. I think such a good talent and then very pleasing personality, and who does, I think, I see her after you. She's doing, who does things exactly in a similar way like you, and without any delay. I think we must bring her home, sweet home.
I don't want to go through.
No, I think, sir, well said. I think, Aparna is always mesmerizing, always positive, always smiling, pleasing, and always willing to help, people. And also, she's a doyen of Bioclues. I would always say that ever since she joined Bioclues, I think she has been the queen of Bioclues. But my only worry is Aparna would not come because now she has a pet. She has a pet cat.
No,
I don't know.
she has international trip, she's ready to go actually.
No, that would be wonderful, Aparna. I think you should bring her along as well. I think
Yes, I will
India,
go back. If I go back, I will go back with her, definitely.
there is a wonderful brain drain going on in India. I think India needs wonderful talents like you. We are quite honored. She has also been an associate editor managing the submissions for Bioclues Journal IJCB, with Jaraman sir, who is the editor in chief. I think before I give one final word of thanks, you should also know something about Aparna. In fact, we are so-called, the core members. So far, we have had about 160 core members, 163 core members. I think she just joined about 1 year ago, 1 year, 3 months ago, and ever since she joined, I think she has been mesmerizing. She has done all the tasks, and she has been very much helping, and we are quite ecstatic and honored to have Aparna Banerjee in our core team. And I can openly say that she can even become the president of Bioclues in the near future. Thank you so much, Aparna, for your kind words and for your kind helping and all through.
Thank you so
I
much,
think
Prash.
Jayaraman sir will also agree to this. Thank you so much. Yeah.
Thank you so much. Have a good day, everyone.
Yeah.
A 10% extra, 100 plus 10.
Yeah, you see how Jayaraman sir says, I said only a little, so it's 110% up and now. Thank you so much.
No,
Yeah.
no. Thank you so much.
Yeah. So friends, so this is today. I think you will also go through the recording again. I'm sure I'm definitely going through the recording again in the YouTube page of Bioclues because this is so special because it is Aparna Banerjee. Thank you so much, Aparna. Thank you so much.
Thank you. I have a good day.
Yeah. And maybe we can have Aparna. Did you leave? Yeah, she left. Yeah. So next we have Dr. Prakash Reddy, who is an Associate Director at National Institute of Nutrition, Hyderabad, coming up on November 27th. Thank you so much. Bye bye, all of you. Yeah.